Thank you for joining us in this exploration of plant synthetic biology. This is part of a series of interviews with academic and industry scientists in the run-up to ASPB's first synthetic biology meeting, which is happening August 7th in San Jose. My name's Jenna Gallegos. I'm a postdoc in Jean Picou's lab at Colorado State University and the news editor of the Oxford Journal Synthetic Biology. Joining me today is Dr. Guillaume Barbier with Join Bio. Can you start by telling us a little bit about Join Bio, Guy?
Okay, so Join Bio's goal is to engineer microbes for agricultural solutions. So what we do is take microbes which are in the soil, which already interact with plants naturally, and we try to bring some genetic traits to these microbes for the benefits of the plants.
So my understanding is that Join Bio is kind of a unique collaboration. Can you tell us a little bit more about that?
Yes, it's a joint venture between Bayer and Ginkgo Bioworks.
And can you tell us a little bit about your scientific journey and how it led to being a part of this venture?
Sure. So it all started, so initially I started studying in France. I did a biochemical engineering master in France. From there, I went to the upper peninsula of Michigan in the middle of nowhere to work on environmental biotechnology in a small company called Nitrate Elimination Company. So I thought I would be gone for 6 months. That was about 18 years ago. Fast forward a little bit, I worked at Novozymes for about 8 years. So Novozymes is a Danish company which is mostly known for producing a lot of industrial enzymes, but I never worked on the enzyme expression platforms at Novozymes. I was on metabolic engineering, trying to make another compound which is found in commodity compounds, biosource commodity compounds, instead of something which was based from petroleum. And then at the same time, at the end, when this project was kind of slowing down and we reached all our targets, I started being involved with the BioAg Alliance, which was something done between Novozymes and Monsanto about using microbes for agricultural purposes. And so I got fairly deeply involved into that, and that's where I started being really interested by that field, because removing the chemical from the field to put microbes, I thought was a great idea. And when the opportunity at Join Bio opened, I found that using synthetic biology for these microbial solutions would be really something great and interesting.
There's a lot of overlap between metabolic engineering and synthetic biology. What for you sets synthetic biology apart?
That's a question that I've been struggling trying to understand. I think metabolic engineering is more the end of what you want to do with synthetic biology, and to a point, synthetic biology are the tools to get there.
So what would you say are the biggest challenges for plant synthetic biology as a field moving forward?
I think we still have a lot of work to do in the tool development, so trying to get always cheaper ways to synthesize DNA, having more outputs, more people out there which are synthesizing DNA, because right now it's still a bottleneck.
You've spent a lot of time in industry. Do you have any advice for young scientists who might be getting ready to make the transition from academia to industry?
Yes, I think what really surprised me when I made the switch was how the industry can be organized and how you are part of a team. And that can be a little bit surprising when you're not used, because the world can be cut in different pieces. And it's always what people talk, and it never makes much sense unless you experience it. But the teamwork is something extremely powerful, because you don't have one brain, but you potentially have 10 brains with you.